Hello, everybody. My name is Jordan Massie, and I'm a PhD candidate in the Department of Earth and Environment at Florida International University. And today I'm going to be sharing some of my dissertation research regarding fish migrations in the Florida coastal Everglades. Understanding where, when, and why animals move can be of great ecological importance. And advances in technology like acoustic telemetry have propelled our ability to track these movements. However, questions surrounding how environmental conditions, such as variation in freshwater flows, affect the year-to-year variability in movement patterns have been hindered in part by a lack of long-term data allowing for robust interannual comparisons. Fortunately, as part of the Florida Coastal Everglades LTER, we've been monitoring fish movements in the Shark River for more than 10 years. Here, we draw on these data to examine how environmental variation at different temporal scales shapes the reproductive migrations of common snook, one of the most highly targeted sport fish in Florida. Snook are widely distributed throughout freshwater and estuarine habitats, but as marine obligate spawners must move to coastal inlets and marine areas to reproduce. We use a two-pronged modeling approach based on the conceptual framework depicted in the center of the poster to ask, one, how do conditions experienced prior to the spawning season, or migratory primers, influence the decision for individual snook to migrate or not to migrate each year? And two, do specific environmental cues trigger the timing of migration within a spawning season? Analysis of our acoustic telemetry data revealed wide variation in both the proportion of fish that migrate annually and the specific timing of migration within the spawning season, which is depicted here in Figure 4 of the center panel. Further, we found that freshwater flow characteristics acting at different scales interact to shape the annual migratory response. Our models suggest that the proportion of snook that migrate each year is influenced by fish size and seasonal drawdowns in water level, which affect the concentration of freshwater prey entering the river channel from drying marshes, and that the timing of migration is largely explained by high flow events resulting in increased water levels and high daily rates of change. Climate change, restoration, and water management will all impact the timing and magnitude of freshwater flow pulses in the future. And by anticipating how snook might respond to these changes, we can inform conservation and management efforts aimed at providing conditions which maintain the environmental dependencies of this recreationally, economically, and ecologically important species.